Good morning, everyone. Myself, Mr. Lovekesh, Assistant Professor, Department of Pharmacology from ISF College of Pharmacy, Moga. And today I am here for the practical demonstration of the B.Pharm 6th semester students for the Pharmacology 3. And the subject code of this practical session is BP608P. And today we will discuss the virtual experiment demonstration, that is to study the anti-ulcer activity of a drug using pylorous ligation method. And in this method, we will ligate the pylorous part of the stomach. So now I will demonstrate that what are the different parts of the stomach. So in this, during the anatomy and physiology, we have clearly seen that our stomach is a J-shaped structure. So in this, there are three main parts. This part is known as the fundus part. And this part is the body. And this part is the pylorus. And this part is the cardiac. So this is the basic anatomy of the stomach. So in this experiment, we study the anti-ulcer activity of the drug. So in this experiment, we will ligate the pylorous part of the rat. In this, we are using the rats known as the rat. So in this, we will ligate the pylorous part of the stomach. In this peptic ulcer, in the peptic ulcer disease, we all know that it's a gastrointestinal disorder. In this, there is an imbalance between aggressive and protective factors. And in this, there is an accumulation of gastric juices in the stomach. So in this experiment, we will ligate the pylorus part by using the thread. And accordingly, there is accumulation of the gastric volume in the stomach. And by the passing of days or hours, you will see that in the inner lining of the stomach, there is a formation of ulcers. And in this experiment, me and my students will practically demonstrate the today's aim. And here is my team.
My name is Muskan Sanan, a student of B.Pharm 6th semester from batch R. My name is Kushman Kaur from B.Pharm 6th semester batch R.
I am Anubhav Arora from B.Pharm 6th batch R.
So now we will practically demonstrate the today's experiment, that is to study the anti-ulcer activity of a drug using pylorous ligand method. In this, we will use the software known as Ex-Pharm software. So this is the first page of the Ex-Pharm software, and now we are using the experiment that is to study the anti-ulcer activity of drug using pylorous ligation method. So this is the first page of the experiment. In this, you will see that two questions over there. One is green color, one is for virtual part. And we just click on the virtual practice. And there is a link created over there, and we just click over it. So this is the first page. In this experiment, there is a scientist in the 1945, his name is Mr. Shea, that introduced this method. In this method, we will anesthetize the rat, and after anesthesia, then we will, before the method will be introduced by the scientist Mr. Shea in the 1945. In this, the pylorus part has been ligated of the rat, and after that, there is accumulation of the acid secretion in the stomach. And after some hours, around 19 to 24 hours, there is an onset of ulcers on the inner lining of the stomach. Now we will just click on the experiment part of this. So this is the instructions for performing the experiment. In this, rats have been fasted for 24 hours prior to the experiment. And randomly, the animal is divided into two groups, as I have already mentioned, vehicle and drug treatment. And in each group, there is three animals. Drug administered once for 2 days and 30 minutes to the ligation. Rats anesthetized with the ether. Pyloric ligation procedure has been done in this experiment. Rats placed in separate cages and allowed to recover. If we are not separating the animals, then there is a chances of that there is an internal fight between the different groups of animals. And after 19 hours, the pylorous ligation animal will be sacrificed by the decapitation method. And abdomen will be the open and stomach has been dissected out from the rat. And after that, contents of the stomach will be collected in the centrifuge tube and stomach opened along greater curvature and ulcers observed under the 10x magnification. Just click over the cross. And this is the window and just click over the administered part. And this is the video how we will perform. So in this anesthesia will be given to the rat. And we will put the incision cut over the abdominal cavity in the midline. And we will find out the stomach. And now we have find out the stomach part. This is the pylorous part. And with the help of the thread, we will tie the pylorus part. We will tie the pylorus part like this, that there is no stoppage of any blood supply. And if we stop the blood supply, then there is a chance of death. So we clearly tied the thread over the pylorus part. And after that, we will perform the suture process to cover up all the surgery part. So now, rats will be divided into two groups. One is the vehicle, one is the drug treatment. And now my student will perform this experiment according to the group. Now first one is this Muskan Sanam. Please come over here.
Okay. Now this is the first group of animals that is vehicle treated. We have taken 3 rats. As you can see in the first rat that is vehicle treated, a midline abdominal incision has been made and the stomach part is being dissected from it. Now this stomach part is being fixed using the pins on the paperboard. can see, it clearly depicts that 3 ulcers has been there in the pyloric part. Now what we'll be doing, this 3 count has been recorded here. Similarly, moving ahead to the next track, following the same procedure, unlike the previous rat, this rat is having 2 ulcers. Now recording its count. Now I would be handing over this experiment to my friend Gursimran. Now moving towards the last rat of this group, performing the same procedure. As you can see, there are total 2 ulcers formed, recording them in the table. Now we can move towards the drug-treated group, where we have similarly 3 rats. In this group, we have administered a miprazole drug, doing the same procedure as we have done in control, or we can say treated group, vehicle treated group. Now we can see there are total 1 ulcer is formed in the treated group, recording in the table. Now I am going to hand over this procedure to my friend Anubhav.
So moving ahead to the next rat, by following the same procedure. So we will see the number of ulcers are reduced by moving ahead to the last thread. So again we will see the ulcers are reduced to 1.
So after performing this experiment, now we just go to the observation part. So as my students have performed that experiment, the reading of the count of the ulcer has been reduced in the case of omeprazole treatment. And this is the observation table, and you will see there is a student name. After that, just click the name of the student. For example, I'm typing Muskan. Then just download the observation table. And this table, you can take the printout of this table and place over the practical file of yours. And now we will go to the conclusion part. So by this experiment, we have clearly seen that the number of counts of ulcer has been reduced in the case of omeprazole. So this is the last part of the experiment. I hope so. All students will have gathered the proper information regarding the anti-ulcer activity of a drug using pylorous ligation. Thank you so much.